Thomson Reuters list of the world's most influential scientific minds in the fields of psychiatry and psychology. He has been an innovator in many different aspects, from his early days studying non-psychotic relatives of schizophrenia patients, in which he found structural brain abnormalities and neurocognitive and social abnormalities that were influenced by the subject's genetic loading for schizophrenia, but were not influenced by medication, etc. He has really led the field in understanding the validity of adult ADHD, did the first family studies of the disorder, and was really a key investigator in showing that ADHD persisted from childhood into adulthood, and showing that about 2 to 5% of the adult population actually has ADHD. I came to know Steve in around 2003 or 2004, when he was the leader of the field of ADHD genetics, and I must say that he really inspired me, as well as many other young researchers, or in my case, formerly young researchers, in the way he conducted research. Steve was really early on convinced that ADHD genetics is team science, and he did something about it and founded the ADHD Molecular Genetics Network, which brought together over many years the leading scientists in this field. In doing this, he showed an amazing talent in building sustainable collaborations. He led the group, entirely unselfish and supportive of others, totally lacking the big ego that he would be entitled to, given all the accomplishments that he has. Many subsequent collaborations were born in this ADHD Molecular Genetics Network, like our own Adult ADHD Impact Group and also the Multicenter Image Consortium. The Molecular Genetics Network also laid the groundwork for the formation of the Psychiatric Genomics Consortium ADHD Working Group, which Steve leads until today in this same unselfish, dedicated and supportive way he has shown earlier. So I think this is enough of introduction. Give the word to Steve for his download.
I think the rest can only be downhill after that very nice introduction. So thanks very much, Barbara. I appreciate that. It just proves that I've been very hyperactive in my career, so I know the phenotype of ADHD very well. We're going to talk about a lot of things today. We'll start with for those of you who don't know ADHD, a quick overview of epidemiology, neurobiology, genetics, the phenotypes, so you can understand that a little bit at least. Talk about some of the new genetic data about ADHD, a little bit, not a lot. I'd like to talk more about some of the new molecular pathways that are implicated in ADHD, how these might be related to areas we think are important in disorder, like difficulties in synaptic plasticity, associated with learning memory deficits. Talk about neurogenesis in ADHD, and that might be a key pathway for the disorder, a little bit of oxidative stress, or a lot actually. I'll end up talking about dopamine, and for those of you who have been in the field for a while, dopamine is sort of the old ADHD theory. It's been around for quite a long time. Some of the newer pathways aren't directly supportive of that. Yeah. I'm sorry, is the mic on?
Yeah.
Can you folks hear me up top? Yeah. Yes? Okay, so ADHD, a chronic impairing childhood disorder, main symptoms: inattention, hyperactivity, impulsivity. Prevalence is about 5% worldwide, and it doesn't differ statistically around the world, except for there's a little bit of an excess apparently in Africa for some reason, and a little bit of a decrease in the Middle East. But there aren't many studies from those areas, so even though it is statistically significant, but we're not sure why. But Europe, America, Scandinavia, Australia, very much the same prevalence. It's more common in males than females, and no one's ever really figured out why that's the case. So that's about the ratio in this population is about 3 to 1, in clinics it's about 9 to 1. It persists into adulthood in about 2/3 of cases, and importantly, it's associated with a lot of impairments. I put a few up there. I think I didn't put up suicide, which is one of them. Criminality is another one. ADHD gets a lot of attention in the news media as sometimes an invention of American society or American culture. It's not really a disorder. It actually is a disorder with very serious consequences. This is the history of ADHD, and you can read this paper if you want to go into some details. It's a review paper I did with Professor Franca, Budelar, and some other of our ADHD expert colleagues around the world. And I only showed to point out that the first description of ADHD in a medical setting textbook was in a German textbook in about 1775. Another one in the Scottish textbook at the end of the 18th century. So the disorder has actually been around with us for quite a long time. It's changed names. The description's changed. It's become more reliable in the hands of psychiatrists. But it's not a new disorder. It's nothing that's new with modern culture. It's been around for quite a while. In fact, the earliest mention here is 76 in the textbook. I ran across this, of all places, in the Scottish National Gallery. This is a Dutch painter, Jan Steen, who in 1670 painted this painting. This is actually an excerpt from a painting that's called School for Boys and Girls. And it has all the things we need to know about ADHD. You got your inattentive boys who can't focus on their work because they're looking over here. You got your hyperactive impulsive ones who are totally acting out and being foolish. These are the kids who don't have ADHD. And this guy might have what we call sluggish cognitive tempo here at the bottom. he's falling asleep. So again, ADHD has been around for a long time. This might be the earliest demonstration of it, although I imagine if you went back further, you'd find even more examples. So genetics. The reason that I got into genetics of ADHD and many other people is that the data from twin studies are very compelling. These are about 20 twin studies from all around, well, not all around the world, but US, Scandinavia, Europe, and Australia. And they've measured ADHD in different ways, grading scales or structured interviews or psychiatrist interviews, but no matter where you find it and where you measure it or how you measure it, the disorder is highly heritable, the average being about 0.74, which is about as high as bipolar disorder and schizophrenia. So it is a highly heritable disorder, and that has launched the whole molecular genetics initiative that Barbara mentioned into ADHD. There are also environmental risks. These are much harder to establish because they require long-term studies, prospective studies, which there aren't a lot of. Some of these data are based more on retrospective reports, and there'll be some debates among people about should some things be on this list or not. We do know that many of the exposures that seem to be associated with ADHD occur very early in life, so they're fetal exposures. Exposure to ethanol, constituents of cigarette smoke, polyfluorinated biphenyls, and lead are perhaps the strongest findings that we have. Along with actually premature birth, low birth weight, and anoxia, that cluster of exposures that occur very early in life have also been fairly consistently associated with either ADHD or ADHD syndromes, inattention, hyperactivity, and impulsivity. Food colorants, less certain, but there's some good experimental data suggesting that food colorants, and if someone knows why they might cause ADHD, please tell me, because I'm really interested in trying to figure out that connection, but I haven't quite figured that out yet. Early deprivation in some of the Romanian adoption studies where children were just left in cribs and not attended to, given no social stimulation. They essentially were just fed and given nothing else. These kids, when they're growing up, have many problems. One of them is ADHD as well. Mild traumatic brain injury later in life. We recently published a meta-analysis on this showing that mild TBI predicts subsequent ADHD syndrome. And then some questionable ones at the bottom: diet, pesticides, air pollution. We're not quite sure about, but have been recently in the literature as potential causes of disorder. So we're talking about disorder, multiple genetic, environmental risks. I can briefly go through what we know about what we decided, we meaning the group of us that wrote this paper, about the brain and ADHD. These are well-known executive function circuits in the brain. These were the first circuits that have been of interest in ADHD, primarily because the first people studying it saw ADHD as a syndrome of disinhibition, and were making comparisons to frontal lobe syndromes. Subsequently, structural MRI studies, functional MRI studies, DTI studies have all confirmed that in these brain regions you either are seeing smaller structures, reduced connectivity, changes in activation patterns compared to non-ADHD people. So there's very strong evidence that these executive control circuits are involved in ADHD, and they do explain some of the disorder's phenotype: disinhibition, inability for planning, difficulties with abstraction, temporal sequencing, time management, and so forth. So these top-down executive control is, of course, a problem, but also the reward circuits are equally clearly implicated in ADHD. Although these came later in, these studies tended to come later in the history of ADHD. We now know that ADHD people show clear deficits in reward processing. They're less sensitive to rewards. They show impairments in delayed discounting, meaning they're less sensitive to more distal rewards, which makes it hard to plan because they're more kind of glued to the moment of current rewards. And the circuits that are involved in these reward circuits are the same circuits that are involved in substance abuse disorders, and ADHD people are high risk for those disorders as well. So we think that this constellation of reward deficits explains that comorbidity. I mentioned initially dopamine as being one of the old friend of ADHD researcher. The circuits we talked about just in the prior two slides are mediated in part by dopaminergic and noradrenergic circuits. Again, these are well-known circuits in the brain. Why have they been studied? Well, they've primarily been studied because of the neuropsychopharmacology of the disorder. As many of you in psychiatry know, theories about disorders tend to get developed by the drugs that work well for those disorders. ADHD is no exception. The medications that treat the disorder either work in the dopaminergic circuits or noradrenergic circuits. The stimulants, methylphenidate, and amphetamines block the dopamine transporter and have a remarkable effect. I mean, they begin to work in about 2-3 days. You see an initial effect. Within a few weeks, some patients become extremely well normalized. Other patients are doing very well. The noradrenergic drugs don't work quite as well, but they do have a substantial effect on the disorder, either by blocking the norepinephrine transporter or by working as an agonist at the alpha-2 adrenergic receptor. Because the dopamine transporter blockers have been the most effective treatments, those have played a central role in most theories about ADHD. In fact, in this next slide, I will show you is people that have done in vivo imaging of the dopamine transporter have shown, for the most part, and we can see that under these studies here, you can't see that down too well, but these studies here, the 0 means no effect. This is ADHD greater than comparison. This is comparisons with less greater than ADHD. Most of the studies are showing a significant effect. These two studies at the bottom don't, but the red diamond at the bottom indicates that overall there's a significant excess of dopamine transporter density in the brains of ADHD adults. The two negative studies, we could bring people here who did these studies and have lots of arguments about who's right. The two negative studies do have some flaws in terms of, for example, not screening for nicotine use and things of that sort that might change the results. So again, we're going to get back to the dopamine transporter later in the talk, but highlighting here just in the beginning, so people who don't know ADHD know this has been a key protein in studies of the disorder. I call this the new genetics of ADHD because we recently broke open the new results from our genome-wide association study that we've been working on for quite a long time, mostly with negative results for at least a decade of meetings where we had nothing useful to report. But now we have actually some very nice results. I've had the privilege of coordinating this consortium, which now has a much more complicated name because a lot of institutions are involved. This is more, the kind of official name now is becoming Psychiatric Genomics Consortium, ADHD, eyesight, SSSI, broad collaboration. And the reason there is because there's been a very big contribution from the Danish group known as eyesight and their collaborating institution, SSI, I forget what that stands for, which has provided many, many samples to this consortium. As you can see, it's a worldwide consortium, contributions from many sites and many colleagues. It's a decade of work, and it's summarized in just a few slides. This first slide doesn't cover all the data we have right now because we haven't processed all the new CNV data. CNV's copy number variants are moderately large insertions and deletions in the genome that are typically rare, by rare maybe 1 in 1,000 or certainly less than 1 in 100, that are usually pretty interesting because they usually involve a gene very directly, like for example they might cut out half of a glutamate receptor or a nicotinic acetylcholine receptor is one of the stronger findings. Summary of this graph is that almost every study that has published their CNV findings finds an excess burden of CNVs, meaning more cognitive variance in ADHD versus controls, except for the second study listed there, the Canadian study, did not find that. But as a group pulled together, they did show very significant excess of CNVs in ADHD. And we're hoping to replicate that in our new sample. This is a new sample, which is much bigger now, with almost 20,000 cases and 30,000 controls, which by GWAS standards in psychiatry is now still somewhat small, and we need to have probably 100,000 cases and controls to really find the bulk of the genes for ADHD. For those of you not used to seeing this plot, the horizontal line around 8 in the middle is the line of genome-wide significance, and any of the each of the dots on the plot is one SNP or single nucleotide polymorphism. It's marking a place on the genome. If that SNP is over the line, it means it's genome-wide significant. It's called a Manhattan plot because they typically, when you have a lot of significance, the plot it resembles the towers of Manhattan and skyscrapers. So that when you see significance, you want to see, like on the left, for example, a very tall building. Because all those SNPs are not tagging different genes, they're all basically the same genetic locus tagging the same gene. Of the ones that are above the line, we're currently convinced that at least 8 of them are genome-wide significant loci. 2 of them we're not so sure about for technical reasons. So the good news is we have 8 new genome-wide significant loci. I'm not going to talk about all these today, but I'll mention the one that perhaps the field is most excited about, which is the FOXP2 mutation. It's been shown now to influence particularly speech and language deficits in a pretty influential paper that appeared in Nature a bunch of years ago. Since then, there have been a number of phenotypes associated with FOXP2 mutations that are reputatively related to ADHD. Speech and language deficits are not the cardinal symptoms of ADHD, but they do occur in the disorder. About 20% of children, 20-30% of children. There's other studies show abnormal activations and positive strong emission tomography studies. Some studies show reduced methylation in the caudate nucleus, which is interesting because that's one of the findings in the ADHD neuroimaging literature, is reduced volume in the caudate nucleus. Although that's apparently only in clinically referred samples, it has not been seen in the general population. The finding about intellectual disability is interesting because ADHD people they don't have intellectual disability, but they do tend to have IQs that are about, on average, 9 points lower than the general population, so there's an effect on overall intellectual functioning. And certainly cognitive and motor delays are a common sign of ADHD kids. So we were quite happy to see the FOXP gene jump out as literally the significant SNP is right on top of that gene, and we're fairly convinced that this is the gene that is tagging because it's these prior associations. I'm not going to talk about some of the other genes. We'll talk a little bit about pathways in a minute. This is a very simple way to kind of think about how it would parse the etiology of ADHD. Based upon twin studies, what we know about the common SNPs. Now, the common SNPs, which I haven't shown the data, but we have data showing that there's, in addition to our genome-wide significant SNPs, there's a very large polygenic background, which may be thousands of loci involved in this polygenic background that account for about 1/3 of ADHD's heritability. And just kind of putting all that data together, about a quarter of ADHD's, of the etiology of ADHD we can attribute to either the environment or error. By that I mean things we can't measure, so we're not really sure what it is. A little bit more than a quarter we can attribute to now attribute to common variation, common SNPs that we've seen in the polygenic score, polygenic background of ADHD. And then the rest, which is almost half, we're not really sure what it is. It's either the rare variants, of course, going there, gene-environment interaction is part of it, and possibly the borderline between, this is not, I think, not well defined, borderline between the blue and the gray here, not defined. We may, in subsequent bigger samples, show more of a contribution to common variants. That remains to be seen. There's a lot that's unknown about where the contribution of rare variants starts and gene-environment interaction begins in terms of that gray area of work. So gene-environment interaction being one of the more difficult things to study in this disorder because the environmental exposures typically occur early in life, but we have the patients later in life, and we can't measure those exposures directly. Okay, so let's talk about, I'm going to talk about two molecular pathways that I've been interested in lately, partly deriving from genome-wide association data, partly deriving from other areas of work in ADHD. So this one is, the neurogenesis and neurite outgrowth is kind of interesting because the Radboud group, Brett Perlmanzer was here, and his Paul Gerhardt, I forget your last name, I'm sorry, who had done this wonderful work in what they call developing genomic landscapes, published this paper in 2011. So this is based on a very small, relatively small GWAS sample. And I was telling Lynn this morning, or telling Gerhardt this morning, that when the paper came out, I was very skeptical. I thought it was based on not enough data, and I didn't understand the methodology because I don't think I'm smart enough to figure that out. It turns out that this group was right, that this early presentation of these data was right in the sense that we've seen that with some of the genes that are, some of the top genes in the genome-wide association study are also implicating this pathway. I'll talk just a little bit about that. One of them is semaphore and 6D. This is clearly involved in neurogenesis in the brain, regulates axon guidance, involved in the modeling of synapses. There's no doubt that this is, I'm not sure if it was in the original landscape, but it certainly is related to the landscape picture that you saw in that original paper in the American Journal of Psychiatry. And then I've already mentioned FOXP2. FOXP2 is, well, first of all, it has clear brain localization. This is something I just pulled off of ingenuity pathway analysis very quickly. But it's located in areas that are very central to ADHD: striatum, cerebellum vermis, BTA, thalamus, and a number of other areas. It's an interesting gene because if you look at the transcription factor binding sites in the genome, there's bioinformatics data suggests that it could bind directly to up to 300 to 400 genes in the human genome. So its regulatory capacity in the human genome is really immense. In this study of transcriptional targets of FOXP2, it was very clear that the gene is regulating neurite outgrowth, morphology of axons, and essentially growth cone dynamics during neurogenesis. So as neurons are seeking out their targets during neurogenesis, this gene is playing an important role in that process. So clearly, the pathways that were talked about in the original Perlman's paper are now being implicated by this gene. And this is just another view of FOXP2 from the gene expression study. If you look at modulations of other genes based upon changes in FOXP2 in the mouse brain and mouse brain, you see these are the pathways that come up as the most important pathways. All very consistent with this idea that FOXP2 and likely many other genes in ADHD polygenic background are regulating events that occur very early in the brain. Okay, so summary very quickly of this particular pathway. I think what's most interesting about the neurite outgrowth pathway is that it could explain why the ADHD brain develops in an abnormal way to begin with. So why, if you will, during the fetal period, the brain is rewired in a way to be susceptible to ADHD, which then onsets, the disorder then onsets at a variable age. We don't know exactly why that happens, but the substrate is set very early in life. But that also, the same pathways are also involved in remodeling synapses and the types of events during synaptic plasticity that take place during learning and memory. And so they could explain why ADHD people also have learning and memory deficits throughout their lives, as long as they have the disorder. So, second pathway that I've become interested in is oxidative stress. We've been interested in a few directions. For those of you, imagine most of you know that oxidative stress, but quick overview. You may have all heard of the so-called oxygen paradox. We need oxygen to survive, and yet the process of creating energy in the cell. Oxygen takes on an extra electron from the electron transport chain, becomes essentially very toxic to proteins, lipids, carbohydrates, and even DNA. And if there's too many of these reactive oxygen species in the cell, you get lots of cellular damage, cell death, and atrophy in brain regions. It's been very heavily studied in the areas of neurodegeneration, where it's shown to play a role. For example, Alzheimer's disease and Parkinson's disease, much less in ADHD. It has been, it's relatively new in the ADHD area. Brain very susceptible to oxidative stress because it has high oxygen use. The antioxidant defenses aren't great, and it has high content of lipids. So why would I bring up oxidative stress? It's actually not a common, if you read reviews of ADHD, you're not going to see mention of oxidative stress in ADHD. One reason is that looking through the environmental risk factors for ADHD, the ones that I've highlighted here in red, this is the same slide as before, but the ones that are highlighted in red are all exposures that have been shown in typically experimental kind of paradigms in vitro or animal studies to lead to increased oxidative stress. And we also know they're associated with ADHD. Correlational evidence, but it's kind of intriguing. Next intriguing result came from the pathway analyses that came out of our cognitive invariant data that I showed you before. This is a paper that's pressed now in molecular psychiatry. The highlighted a few pathways in color that are, the green pathways are immune system, the red pathways are ion channels, but the ones that I'm interested in now are the ones that are kind of the brown pathways. There are both pathways involved in the electron transport chain, which is mitochondrial functions that are involved in eventually creating oxidative stress. So again, more, I would call modest evidence suggesting that perhaps oxidative stress pathways, mitochondrial dysfunction might be involved in ADHD. So then I had reviewed the literature recently on, I was interested in non-pharmacological treatments for ADHD. For those of you who are interested in that, we've published a book recently on the topic. One of the more recent findings in that area has to do with omega-3 fatty acids. There are now 2 meta-analyses published which show that omega-3 fatty acids, which have many positive effects, one of which is an antioxidant effect, do reduce the symptoms of ADHD. Now, it's not recommended as a first-line treatment for the disorder because the symptom reduction is not dramatic. It's about a quarter to a third of what you'd see for example, stimulant medication, but it's nonetheless significant and real. Another observation that I had made in my university, a colleague of mine is a lupus researcher, and we were talking about lupus, and he mentioned to me that many of his patients seem to be very inattentive. So we did some studies of his lupus patients. Now, lupus is a disorder which is known to be associated with high levels of oxidative stress. And he was already treating them with N-acetylcysteine, which is a very potent antioxidant. So I said, well, why don't we measure their ADHD symptoms in your group? And we did. We found out that, number one, that the lupus patients did have, these adults with lupus had high levels of ADHD symptoms, particularly inattentive symptoms, and that in the treatment study of N-acetylcysteine, there was that N-acetylcysteine significantly reduced lupus symptoms, but also significantly reduced the ADHD symptoms, particularly in dementia. So again, possibly, the idea that antioxidants can treat ADHD derives from these studies of lupus. There's also a very small literature on zinc deficiency in ADHD. And I mean, zinc deficiency, I mean severe zinc deficiency, which usually doesn't occur in the West. For some reason, there's a series of papers from Iran where there's, for some reason, a high level of zinc deficiency. And in these Iranian papers, they show pretty clearly that zinc deficiency, when treated, number one, zinc deficiency seems to cause ADHD, and then when you supplement these children with zinc, the ADHD goes away. Possibly, again, this could be another indicator of an antioxidant helping these kids with zinc deficiency. And there's this new paper that just literally came out. I just found it a few weeks ago. It's a group that had been studying peripheral levels of oxidative stress in ADHD, really in the blood of ADHD kids. And they did this, they showed high levels of oxidative stress measures in the periphery. And they showed when they treated the kids with methylphenidate, of course their ADHD got better, but also the measures of oxidative stress also reduced. And so they were claiming that methylphenidate was having an antioxidant effect. It's unclear to me exactly how it did that. But, so a series of studies suggesting that treatments that treat ADHD and putatively treat oxidative stress also are, may implicate oxidative stress in the disorder. So this is, again, more indirect evidence from some of the work we're doing in my lab at SUNY Upstate. We're talking today that I've had a kind of an obsession with this gene, SLC9A9. It's a sodium hydrogen exchanger that I got interested in a number of years ago because there's a rare variant in an Australian family that causes ADHD. There's another rare variant that causes autism. And then we have a rat model where we have mutations in the gene that lead to an inattentive rat that has led us to kind of look at this gene and the potential relevance to ADHD. It's involved in endosomal trafficking, and because it's trafficking proteins to and from the membrane, it is involved in, for example, the recycling of the dopamine transporter to the membrane. Another reason we thought to be involved in ADHD. So we published a bunch on this already. This is new data where we've done proteomic studies trying to map out the proteome of the gene, and then looking at the proteome of the wild type, meaning the normal gene on the left, and on the right side, one of the ADHD mutations. Now this is hard to process because it's all these colors and balls and so forth. Essentially, each little circle is a molecule, the protein, and the color of the node is corresponding to how close it is to other nodes, and the size corresponds to its number of connections. Essentially, it's a visual picture of how the mutation changes the topology of the network. And all we need to know for right now is that the topology does change with the mutations. So what we were interested in saying is, well, if we look at the changes in the protein, the changes from here to here, which are hard to see, but they're there. If we take those changes, we quantify that, we define interactions that are changed and those that are not, and let's look at which biological pathways are implicated in those changes, we get these results. And these results give both the ADHD mutation in black and then the autism mutation. Autism mutation is in that white and black hatched lines. As you can see, the first and the third are both involved in oxidative stress pathways. First, mitochondrial dysfunction. Third, oxidative phosphorylation. And these are all highly significant. These are corrected for multiple comparisons after looking at lots of different canonical pathways. The title of the graph is NHE9 because that's the protein is called NHE9, sodium hydrogen exchanger number 9. So we're beginning to think that SLC9A9 is involved in oxidative. We don't know why it's involved in oxidative stress. We think it's probably involved in the trafficking of proteins that have been, if you will, destroyed or altered or damaged by oxidative stress, and therefore it becomes involved in this process of cleaning out the cell from these damaged proteins. I mentioned that there was one study that looked at methylphenidate and personal levels of oxidative stress in ADHD. There are a number of studies that have actually now looked at oxidative stress measures in ADHD in children and adolescents. This was a meta-analysis that we looked at two things. At the very top, these are measures of antioxidant capacity in the periphery. So it's the first half of the graph, and then the bottom half of the graph are measures of oxidative stress. So when we looked at the antioxidant measures, there was no difference in antioxidant status between ADHD and controls at all. But when we looked at the measures of oxidative stress, the ADHD group was significantly higher risk for having higher levels of oxidative stress measures in the periphery than were the non-ADHD kids. This was across most of the studies. I think there were 3 studies that disagreed with the finding, but as a group, the studies in the second half of the slide are consistent with this result. So this got us more thinking about what directions we could take the research. Recently, my group has been trying to decide on what directions to take this research into. One of the things we showed in the meta-analysis is the effect of age. And age in ADHD is kind of interesting. We'll talk more about it later because there's age-dependent decline of the disorder. We found here that the association between ADHD and oxidative stress was greatest for children than it was for adolescents. And we also found only one study in adults which found no association, which led to the hypothesis, and I will always kind of put in this brown color something which is just a hypothesis that I have and is not data that anybody has shown. It's possibly that the re-regulation of oxidative stress pathways during adolescence and adulthood accounts for some of the remission of ADHD. Synapses are very vulnerable to oxidative stress, and I'm beginning to think that in ADHD, one of the puzzles that the oxidative stress findings give us is that most of the disorders that have been studied that are related to oxidative stress cause extreme neurodegeneration, which is not with ADHD. And I think it may be the specific sensitivity of the synapse oxidative stress to lower levels of oxidative stress that may be involved in ADHD. The data that these folks had published, this is actually a review paper of antioxidants and redox signaling. They particularly noted that the promoters of a specific sets of genes that were synaptic plasticity genes were selectively damaged by oxidative stress. They weren't able to explain that finding, but it does seem that there's a extra sensitivity to synaptic plasticity genes to oxidative stress that's occurring at the synapse. This is what I was looking for. So we talk about oxidative stress as a problem, and in fact, yes, at very high extreme levels, oxidative stress causes extreme damage. This is a cartoon that these authors provided of saying that when oxidative stress becomes too strong, the damage that it does to the cell outweighs the positive effects it has on the signaling and synaptic plasticity. We think of reactive oxygen species as bad, but actually they're used in signaling networks in the normal everyday life of the cell. We generate the reactive oxygen species. These communicate to the membrane. They have effects on the recycling of receptors and transporters based upon the energy needs of the cell. What happens at physiological levels is that, in the cartoon, I guess, here is just showing that at the physiological levels, the good effects of oxidative stress are outweighing the bad effects of oxidative stress. And we're getting enhanced synaptic plasticity, enhanced LTP, enhanced long-term memory. The hypothesis that we're working on from this is that there are moderate levels of ADHD associated with oxidative stress that are impairing the synapse to moderate degrees, and I say moderate compared to, for example, Alzheimer's disease, that are explaining the ADHD's deficits in learning and memory, and also its dysregulation in neurogenesis. That there's a fight, it's a battle in the cell, if you will, that it's because of some dysregulation of oxidative stress pathways, it's trying to re-regulate to having more normal physiological levels, but it's not getting quite to that level, and that's leading to these to damages, a mild level of damage that's causing the ADHD symptoms. I was talking about damage to the brain, so I'm going to just talk about a little bit about brain structures in ADHD. I'll show this slide that Martina Hoodman put together. Well, I put the slide together. This is data from Martina Hoodman's paper on from the Enigma Working Group, which is a consortium, a worldwide consortium looking at structural MRI data in ADHD. This is the data from the mega-analysis of 1,700 cases and 1,500 controls. This is the paper that they're working right now. It's under submission, I think, still for the subcortical structures. And this is the pattern of significance of many of the subcortical structures are significantly. This is significantly decreased in ADHD. So the question becomes, how can we relate these decreases in structures to the pathway that we've been talking about today? And we'll talk. We think that some of this might be due to oxidative stress. Talk a little bit more in the next section about how I think that might be the case. Just to summarize this section of oxidative stress, we think that because oxidative stress is damaging synaptic structures, they actually could work in conjunction with the neurite outgrowth pathways we talked about before to impact neurogenesis, leading to the smaller brain structures by impacting early neurodevelopment, and then affecting learning and memory throughout life by having continued effects on synaptic plasticity. how does dopamine come into all this? Because being somebody who, I suppose you get locked into an idea of having studied the disorder for 30 years or so, it's sort of been always in the back of my mind, how do you link dopamine to new data that are coming out? And of course, that could be a mistake. Dopamine might turn out not to be involved at all in the disorder, but we keep searching for links. So one thing I found was this study of looking at dopamine levels in the changes in dopamine levels based upon different FOXP2 mutations in mice. These are two different mutations. The one in black is what's called humanized FOXP2. That's the FOXP2 that carries the version of the gene that's seen in humans, but not in others, non-humans. And the other is just a knockout versus wild type comparison. The horizontal dotted line is just kind of the average expression. 100% expression is average. And essentially what it's showing is that these two mutations dysregulate dopamine levels in different ways. The wild type mutation increases the expression, increases dopamine in these mice. The humanized mutation decreases dopamine expression. The main point here is that there is a connection here between FOXP2 and dopamine in the brain that may be involved early in life, we'll talk about in a second, because of the relationship between dopamine and neurodevelopment. And typically, when the theories of ADHD have always had, for the most part, based on neuropsychopharmacology, been interested in the effects that changes in dopamine are having in the brain of a child that has ADHD at the time they're seen, if you will, in the clinic, and how it affects ADHD symptoms. But dopamine has been shown fairly clearly to have effects in neurodevelopment and neurogenesis very early in brain development. Now, it's a complex pattern. You have activation and inhibition of either D1 and D2-like receptors in the brain, and these are now known to affect the proliferation of neural precursor cells in the developing brain. So dopamine and its receptors are regulating the kinds of effects that we talked about before that you would see from the neuroid outgrowth pathways that you talked about in your American Journal of Psychiatry paper. So this has led me to kind of thinking of a hypothesis about how, to get back to my old friend the dopamine transporter, that how early changes in the dopamine transporter could alter dopamine gradients and receptor activation, and perhaps lead to deviant neurodevelopment during very early stages of development. This is totally a hypothesis. I have no data on this, but it's something that sort of derived from developing this talk and thinking about these new pathways that are involved in ADHD. So my current thinking, oxidative stress leads to overexpression of the dopamine transporter. I'll explain that a little bit in the next slide or two. Perhaps alters dopamine gradients, which may be involved in the axons reaching their targets, leading to changes in neurodevelopment and perhaps the smaller brain structures we see in ADHD. We do know that the dopamine transporter is expressed very early in the brain. This is a download I did from the Allen Brain Atlas. Essentially, it's showing across all brain structures the expression levels of the dopamine transporter, red being higher. So the big red bars there on the right are the situation now you're in the BTA, as you'd expect, to show very high levels. Very early on in the fetal brain, we know the dopamine transporter is there. And the question that I have is really more of a question that we're hoping to solve is if we know that dopamine transporter is dysregulated in the adult brain for ADHD, what happens when it's dysregulated in the fetal brain? Well, so I was surprised to learn, just going through the literature, that there actually is a literature on how a reactive oxygen species affect the dopamine transporter. And some of these species actually bind to the transporter and have been shown to essentially reduce its efficiency. So that we can imagine that very early in life, that when the dopamine transport is being inefficient when it needs to be efficient, the brain is re-regulating itself to make it more effective, which would be to upregulate the transport. And so this is why I propose this theory now, this hypothesis that that oxidative stress impairs the dopamine transporter efficiency, and then there's an overcompensation of the transporter, which helps us, which overshoots when we have high levels of that activity. One of the kind of interesting things that popped out of this inquiry of mine is that estrogen is a well-known antioxidant, and which has been shown to protect against the reduction of dopamine transporter efficiency that we see that's caused by oxidative stress. Whether this accounts for the differences between males and females in the expression of ADHD, I don't know, but it's another hypothesis that I put forward that I think is worthy of testing. Another way that dopamine receptors are involved in brain development has to do with a number of studies now that show that dopamine receptor antagonists can create cascade of autophagy in the postsynaptic cell. Autophagy is a normal process in the cell which is involved in, again, autophagy being self-eating. The cell literally eats its own contents that are the misfolded proteins and organelles that are no longer needed, and then send them to recycling. When autophagy gets out of control, it leads to apoptosis, cell death, and perhaps smaller brain regions. This leads to the idea that if dopamine antagonists can induce autophagy, what happens when the dopamine transporter is upregulated in the brain? Possibly that this also would upregulate autophagy in the postsynaptic neurons, explaining why many of the brain regions that are implicated in having smaller brain structures in ADHD are showing are postsynaptic to dopamine neurons. Some data that are actually puzzling to me and don't fit in well with the theory I have that oxidative stress in ADHD has to do with what are called DAT-TG mice. These are mice that overexpress the dopamine transporter. It's the kind of equivalent to a gene duplication of the dopamine transporter gene. But they do show increased oxidative stress and also a loss of dopamine neurons, which is kind of what you'd expect. This is well known to occur in Parkinson's disease, but it doesn't occur in ADHD. In ADHD, it's not the dopamine neurons that are, it's not the regions of the brain that have dopamine neurons that are showing smaller structures. It's mostly the regions that are postsynaptic to those regions. We also show increased postsynaptic V1 and V2 receptors, as you would expect from not having sufficient dopamine in the synapse. And interestingly, there's increased postsynaptic AKT signaling, which is a signaling pathway that leads back into this autophagy pathway we talked about before. So again, it's the second level of evidence suggesting that autophagy signaling can be dysregulated by changes in the dopamine transporter, which again has led to this hypothesis that a dysregulation in autophagy is accounting for some of these smaller brain structures associated with ADHD in regions that are postsynaptic to dopamine neurons. I showed you this slide before, and I'm showing it again because just to remind you that there's a gradient of effect, and even in these subcortical structures, some are showing stronger effects. This is Cohen's D, which is a measure of how strong the effect is, and some regions are showing smaller effects. So I was interested in how, if you look at these regions, we know that they're innervated by dopamine neurons. Were there differences in the expression of the dopamine transporter? So if we go into the Allen Brain Atlas, and we look at I'm plotting here on the horizontal axis the effect size of ADHD versus control, and then on the vertical axis here, we're plotting the mean expression of the dopamine transporter gene. There's a stepwise increase as the effect size increases, not completely stepwise, but there's a somewhat linear increase between as the effect size increases, we also see an increase in the amount of dopamine transporter expression. That's statistically significant at p equals 0.003. An interesting finding, it's still somewhat preliminary. The Allen Brain Atlas is only 5 brains that have been, they've done a complete mapping of gene expression in many different brain regions. But it's suggestive again that there's something about that the brain regions that are most selectively vulnerable to being smaller in ADHD, tend to have more dopamine transporters expressed in the neurons that are innervated, which would be consistent with dopamine transport having an effect on these brain regions during neurodevelopment. So if this question, which I kind of knew the answer to before I phrased it, so it's a little unfair. So given that increased STAT expression predicts decreases in brain volume instructions, the blockade of the dopamine transporter by stimulant medication, would that lead to larger brain structures? Would stimulants actually be therapeutic for the brain? Well, it turns out no one's ever done that study, as you should do it. You need to do a controlled clinical trial to actually show that. But there are some intriguing data. This is a meta-analysis of structural MRI studies, and what they plotted here on the bottom are each dot is a study, and then what they plot on the bottom are the percentage of patients receiving medications, and then this is plotting the basically it's the size of the plot of the caudate. This is the right caudate here. What they showed for the caudate and a few of the other brain structures is that in the studies that had more patients that had a history of stimulant medication, they showed increases in volume that were more in the direction of the control group, which is data consistent with the idea that people who have had a blockade of their dopamine transporter for a period of time are showing larger brain structures. Again, these data are, they're suggesting a therapeutic effect of stimulants, but they're very naturalistic data, so they're by no means definitive data, but quite interesting nonetheless. So based on all these data, I've been exploring this possibility that dopamine actually might be therapeutic. And it turns out there is a small literature on dopamine in adult neurogenesis. So I honestly don't know a lot about this, but something I've just recently been studying. The regions of the brain in which adult neurogenesis occurs do contain D1-like receptors and D2-like receptors. And there have been a number of experimental studies showing that when you modulate dopamine levels in these areas of the brain, you can change the activity of dopamine. You change the number of progenitor cells that are eventually leading to mature neurons in the brain. The data, the literature on this is very it's small, and it's not always in agreement with one another. So it's suggestive, but it's very consistent with what we've seen in the clinical data I showed you before about the dopamine transporter. This one author was so convinced with these data, she put together this model suggesting that both D2 agonists and D2 antagonists in these experimental studies have differing effects on. You can see there's a lot of question marks in this slide, which is a sign of what we don't know about adult neurogenesis and the effect of dopamine. That there's some effect of D2 agonists on adult neurogenesis signaling pathways that, as you suggested, potentially have a therapeutic potential for she's thinking Parkinson's disease. I'm wondering about this in the context of ADHD, given the data that I showed you already about the effect of stimulants on the brain. So this led me to another question. If the dopamine system is mediating adult neurogenesis, if dopamine blockade is spawning neurogenesis and ADHD-associated brain regions, what is the effect of an age-dependent decline of the dopamine transporter? So you can block the dopamine transporter, or you can lose it naturally because you get older. As we all get older, our dopamine transporter density gets, unfortunately, lower and lower in the brain. There's one example of this this was the first paper on the DAT in ADHD brains in the Lancet in 1999. I'm showing you this because of the red dots are the ADHD. It was only, 6 ADHD people to study the red dots. They all had higher dopamine DAT binding potential. But the bars here, the bar graphs, are the controls. And as you can see, there's this age-dependent decline in the dopamine transporter. So what effect does that have? Is it possible that this age-dependent decline is in part explaining the normalization? I shouldn't say the normalization. I'd like not to use that word. Is explaining the decrease in ADHD symptoms over time from youth to adulthood, which I can show you. This is the review paper I mentioned before. We summarized the data on how ADHD symptoms decline with age. Basically, if you look at the green line, that's showing the decline in people meeting diagnostic criteria. This is longitudinal studies of the disorder, starting with children who are diagnosed and following them up over time. The blue and the yellow lines are talking about changes in decreases in people who would meet clinical criteria for the disorder, meaning that they still maintain ADHD symptoms and signs of functional impairment. That's why we come up with 2/3, about 65% of ADHD kids retain their symptoms, but there's this age-dependent decline, which is curious to me because it may be related to the decline, and it's likely related to the decline of dopamine transporter. And the hypothesis or the question is whether it's somehow related to changes in your brain. Going back to that same structural MRI study, if you look at, again, this study, every dot here is a different study. If you look at the ages of the study, the mean age of the sample that was studied in each study, that's positively correlated with brain matter volume, if you're looking at the data from the right retaining, suggesting that with age, some of these brain structures are getting more like the control group. Some of the decreases in ADHD are getting more like what we see in the control group. There's a series of studies by Philip Shore at the American NIH. He's looking at cortical thickening in ADHD, and showing that over time in longitudinal studies, in some ADHD children, the brain is catching up. I don't believe he's published yet on the effects of stimulant medications, but I would guess if you could look at that, you would find positive effects of the medications. And finally, we're running out of time. I have to mention the dopamine, the famous polymorphism in the dopamine gene that is in the 3' untranslated region. We know from the meta-analysis that Professor Francis published that the 9-repeat allele in this locus associates with ADHD in adults, but there's no association with ADHD in youth. Some people would argue that the 10-repeat allele is associated. I think that's not true. I'll show you why in a second. We showed in another meta-analysis 2 years ago that the 9-repeat allele, the same one that's associated with ADHD in adults, is also associated with increased expression of the dopamine transporter in striatum, in neuroimaging studies. I think it's very likely that what's happening, if I'm right about the effects of oxidative stress on the dopamine transporter leading to decreased neurogenesis, that I think the reason why the 9-repeat allele is in effect in children is that it has a relatively small effect. That early in childhood, that the effect of oxidative stress is too strong, and the 9-repeat allele is not having an effect. But as children age, as the dopamine density decreases with time, there's an interaction of the 9-repeat allele with the age-dependent decline of the dopamine transporter to help to explain why it predicts persistence in adulthood. Again, a hypothesis that hopefully we can test with some of the new data that we'll eventually have from genome-wide studies. And just to show, for those of you interested in dopamine transporter, these are the this is the new GWAS. So if you looked at a big sample of GWAS, this is mostly children, so it's mostly a child sample. There's just no signal at all. Remember, a significant result would be up around 8 here, so there's just no sign at all of significance of the dopamine transporter in this sample, which is a primarily children sample. Okay, I've said a lot today, and I'm frequently accused of giving very dense talks that have a lot of information, and I hope that you've been able to follow this. Trying to develop some new hypotheses for ADHD, and to talk about how some old hypotheses, or not so old hypotheses about neural outgrowth and neurogenesis have been validated by some of the new genetic data. I think that these pathways can actually explain the onset of ADHD's pathophysiology early in life, but also the expression of the disorder during life and the problems that these kids have with learning and memory. What's very interesting to me is that they provide some very new directions for us who are interested in the functional effects of genes. But they're also consistent with a continued central role for dopamine in the etiology and the persistence of the disorder. With that, I will end it. thank you all for your. Thank you all for listening.
Thank you very much for this extremely valuable food for thought. I think we're going home with a lot of questions, but perhaps we can ask a few here as well.